Engineering an enzyme-responsive genetic delivery vehicle. Hello, my name is Jshs, and today I'll be discussing my project on the engineering of a new genetic delivery vehicle at the University of Connecticut Department of Chemistry under Dr. Jessica Rouge. Being from a family of doctors, I've always been interested in new medical technology. When I heard about Dr. Rouge's lab from a friend, I was instantly intrigued by her research in nanoparticles and their medical applications. When I visited her lab and shared my interests, she instantly welcomed me to her group and informed me that she has a new project involving the synthesis of a new type of nanocapsule. I decided to work on this project, and together with Dr. Rouge and my graduate student mentor, Mr. Saket Gudapati, we developed a procedure based on preliminary data conducted by my mentors. During this project, I will be independently conducting all the research under the supervision of Mr. Gudapati, and will develop my own preliminary analysis of data before receiving feedback from my mentors. My research has its foundation in another study conducted by Dr. Rouge's lab based on the nucleic acid nanocapsule. This nanocapsule is made of surfactant molecules with a hydrophilic nitrogen head and a hydrophobic carbon tail. When added to a polar solvent, like water, these surfactant molecules form a structure known as a micelle, which can contain many hydrophobic molecules, such as dyes and drugs, within them. Additionally, thiolated DNA can be bound to the outside of these micelles using click chemistry, allowing the nanocapsules to easily enter cells. One of the most novel developments of this nanocapsule is the cross-linking of its heads. This ensures that the cargo within the nanoparticle cannot be accessed without the presence of a specific enzyme capable of breaking apart the cross-linker. Despite the many capabilities of the nucleic acid nanocapsule, it also has its shortcomings. For example, it can only hold small hydrophobic molecules within it. This is why my project on the reverse micelle is an important expansion. The reverse micelle is very similar to the nucleic acid nanocapsule, except it is flipped inside out, allowing hydrophilic molecules to be stored at its center. If synthesized correctly, DNA the size of entire genes could be fit inside these structures. This could have huge implications in the medical field, as it would be an easy way to deliver correctly functioning genome to specific cells in the body. The challenge is in actually creating this hypothetical reverse micelle. To do this, we have created a preliminary procedure and would use techniques such as dynamic light scattering to determine the size of the nanoparticles, zeta potential measurements to determine their surface charge, and PCR to determine whether the DNA was successfully transferred to the reverse micelle. We can then use these data to engineer a new procedure to test. We expect to be able to successfully synthesize this particle and learn more about its structures and limitations.